information. And if you zoom it in one segment of the dendrite, what you see here are these thorny projections. And they are scientifically called as spines. They are called dendritic spines. And this is where an individual connection is made with the other neuron. Okay? Now we have one of the conundrum in the, so these are called dendritic spines. Now one of the conundrum in neuroscience is that we are born with limited number of cells, around 30 billion. However, even with the same number of cells, we almost have an infinite capacity to process and store information throughout our life. Even when we are 70 or 80, we still have the capacity to learn a new musical language or an instrument, right? Now, how do we achieve that? This is kind of a limitation that we have same number of cells, right? Now, the answer lies in these connections. So one neuron can make up to 1,000 connections. And one of the fascinating properties of these connections, which I will spend the rest of my life studying, is that they are dynamic in nature. They change with experience, okay? And this concept is called synaptic plasticity. Now we can actually measure this live in a behaving animal. If you again focus on one such dendritic shaft, you have these dendritic spines, and you're learning what will happen in that in a specific connection or in a specific synapse, you will have synaptic plasticity or strengthening of synapses. When you're learning, there is strengthening of some specific synapses in simple terms, and when you're forgetting, you are weakening some connections, such as in Alzheimer's or dementia. Now, there are two cool properties here, which I would like you to focus. One is when learning happens, see how quickly the spine volume changes or the synaptic strength is happening. And the other thing is, it is quite synapse specific. It is quite compartmentalized. The other connections are actually not changing. So during this specific learning, whatever it can be, ABC, the decision of whether the information has to be stored is occurring at the synapse at one connection level. Now, this is again not a new observation, but this is something that we recorded in our lab. Scientists have now identified various mechanisms of this type of memory formation. There can be patterns of electrical activity. For example, before the exam, you will be asked to mug a certain concept. What you do is you try to say it out 5 times, 10 times, 15 times, right? So you're repeatedly giving that stimulation. So that can be one way you can change the strength of the synapse. Or there could be release of neuromodulators such as serotonin or dopamine. For example, you suddenly get a call from your crush on a birthday and you get an unanticipated reward, right? And that's when the dopamine is released in your brain, and you'll not forget that easily. So you kind of remember some of those events. And so we have now identified different mechanisms of storing different types of information. Okay? So now this is one concept, that synaptic plasticity happens at the synapse, and this is pretty much happening in all brain regions, and it's conserved across species, from invertebrates all the way to humans. The other concept is now how does this synaptic plasticity happen? Now we know that the brain communicates with each other using electrical signals, right? But the electrical signals or these stimulus are millisecond long. Now how this millisecond long signal gets transduced into something which is long-term storage. So now what happens is there is this beautiful electrical to biochemical transduction that happens, okay? So these millisecond long electrical stimulus can activate, if it's a relevant stimulus, can activate complex biochemical signaling at that synapse. So it could be secondary messengers that last for 5 to 10 seconds. It could be downstream biochemical signaling like kinases and phosphatases, or it could be changes in the gene expression that can really cause changes for 60 minutes, or it could be for even years, right? Now, depending on the type of biochemical signaling, now this is my hypothesis, you'll not find in the textbook, but what we hypothesize is, depending on the type of biochemical signaling, we can have memories that are stored for short term, like for example, you're mugging for an exam and after exam you completely forget that. Or you can have some long-term storage. For example, if I ask you, what did you do for your 18th birthday? Or you can still walk. That's also a non-declarative kind of memory. So for these long-term storage, you have a different type of biochemical signaling. And Again, this is not new. So scientists have identified thousands of proteins that really can play a role in changing the strength of the synapses. I'm just giving you a snapshot of how many proteins have been identified. Okay. So you don't need to, I just need to show the complexity. This is just a presynaptic terminal and a postsynaptic side, and you can just see how many proteins have been involved. Now, the need such complex protein signaling. All we need to do is increase the strength of a synapse or decrease the strength of a synapse. We can do it with 3 proteins. Why do we need thousands of protein to really change different aspects of synaptic transmission? And that is what I start with. Now, why do we need to study it? Why is it relevant? Why will someone give me money to really study these molecular mechanisms of memory? The thing is, these neuronal communications get affected in any major neurological disorder that you can think of. Whether it is Alzheimer's, where we know that the number of synaptic connections actually decreases. This is an image of a healthy neuron. This is just a 2-photon image of a healthy neuron. You see how many dendritic connections there are, synaptic connections, and in Alzheimer's, the synaptic density is reduced. On the other side of the spectrum, you take a developmental disorder such as autism, or Fragile X is a kind of, it comes under the ASD. You see here, now as compared to the healthy neuron, we have a lot more connections than the brain can handle. So understanding the specific molecular mechanisms that really regulate these synaptic plasticity mechanisms, the number of spine density, the number of spines on a dendrite, it will help us to really identify some specific therapeutic targets to objectively treat some of these disorders. But I'll come back to understanding the basic mechanisms of memory. So my goal, or the goal was to really fix this gap right now in really identifying during a particular learning phenomena in the animal, what specific plasticity mechanisms play a role, and underlying those specific plasticity mechanisms, what specific biochemical computation get activated. So it is not the case that every time you'll have thousands of proteins getting activated. So what we hypothesized is that during some forms of learning, some biochemical signals get activated, and some other forms of learning, other signaling cascades get activated. And I try to use different tools to address this, and this is where the genetics part will come in. We use electrophysiology and biosensor imaging tools to understand these molecular mechanisms in the context of a specific form of learning. And this is hippocampus dependent. So one of the cool things in hippocampus, it is a brain region in the medial region, temporal part of your brain. It is responsible for learning and memory and also spatial navigation. This is where your GPS cells are in simple terms. What does that mean? Is when the animal is navigating a new environment, there will be one cell that will be only getting activated in one location. So if you're here, there will be one cell that gets activated. If you move to this part, there will be another cell that gets activated. And so the group of cells together map an internal representation to really make you familiarize with the environment, such that if you have to go from your house to the nearest grocery store, you don't need a Google map, right? You have an internal representation. So you have built some maps or these cells that have represented these location. And so now I can show you here in a video where we are recording from a mouse, and we are doing tetrode recording, so we can really record multiple cells. You'll see that some cells will only get fired. Can someone press? You'll see it's a sped up video, and we have got 10 cells where they will only fire. The pink cell will only fire in this location. The green cell will only fire in this location. The yellow cell will only fire here, and so on. And so together, these 10 cells almost map an entire map of this environment. Okay? And so these cells are called place cells. And we wanted to understand what plasticity mechanisms really underlie these forms of learning. Now, this is individual cells where the learning is happening in the hippocampus. So we can really target these cells and understand the synaptic plasticity mechanisms. Very recently, someone found that a very different form of plasticity, which they termed as behavioral timescale synaptic plasticity, underlies these place cells, where what happens if you record from inside these cells, before these cells became place cells, there would be these large depolarization events or changes in the membrane potential would happen, which would get paired with the synaptic inputs as the animal is navigating the environment, and that potentiates some synapses. And where the depolarization happened, the cell will start to fire or get activated in that location. So we characterized in our postdoctoral work, which is the Nature paper that Parni sir talked about, we characterized this plasticity and understood the molecular mechanisms that underlie that. We asked two questions. Can BTSP be induced at individual synapses? I showed you in the beginning that this can be compartmentalized, this can happen at individual synapses. So we asked whether this plasticity, which is relevant for spatial navigation or place cells, can this also occur at individual synapses? And this we asked using some very complex electrophysiology that I'll show you. And the other is what provides molecular signaling, what molecular signaling gets activated that can provide this behavioral timescale for us to learn this kind of behavior. And here I use some novel Fred biosensor tools. So I use rodent models, mice, and from there I make acute hippocampal slices with which we can really record from cells. We can keep these slices alive for 7 to 8 hours, and we can record from cells of these slices. And using 2-photon imaging, when we are recording from this cell, we can actually fill in a dye. So this is now recording from inside a cell. We can fill in a dye and we can actually look at different parts of a cell while we are recording from this live cell. And we can focus on one small segment of the dendrite where we can see the spine, and using some synthetic compounds such as MNI glutamate, we can record currents from these synapses. Now we are not recording current from a cell or from a brain region, we are recording currents from an individual synapse. Okay, and this is what it looks like. And I use this as my tool to ask the first question. Can I now induce some kind of plasticity to change these currents amplitude? Okay, this is actually not currents, but these are potentials because we are recording it in current clamp. So we found that indeed behavioral timescale plasticity, or the plasticity that is relevant for place cells, can be induced at individual synapses. So what I'm doing here is I'm recording from this synapse and I'm inducing plasticity in this synapse, and you can see that as compared to the precondition, the post showed much larger increase in amplitude. And this is what the normalized EPSP amplitude looks like, where you see only in the spines where we stimulated, we saw an increase in the amplitude overall. And in the spines which we did not induce BTSP, did not show any change. So this is a synapse-specific event. And this is what the magnitude of potentiation looks like. And there is a huge variability in how much the spines potentiate. But this is still overall a significant value as compared to the non-stimulated spine. Okay, so far, it appears that whatever I showed you in the classical plasticity paradigm, it is very similar to the plasticity that happens in during spatial navigation. So what is different? What is novel? Why do we study it? Why did this get published in Nature? What we found is, even though qualitatively speaking, we have a plasticity which is very similar, when we went and understood the molecular signaling that underlies this plasticity, which is behaviorally relevant, we saw something different. Now, to understand molecules, we have heard, or we have found different ways of studying molecular signaling, right? What we can do is we can isolate a protein, we can look at protein expression, or if you want to block a protein activity, we can do pharmacology, we can genetically knock out a protein, right? We can do all of that. But what that tells us is a qualitative yes or no, right? Whether this protein is important for a particular phenomena or it is not important for a particular phenomena. It does not tell us three things, three quantitative information that we were interested in. First, when is the protein getting activated during learning? Where it is getting activated, whether it's getting activated in the synapse or it is getting activated in the whole neuron. Third, for how long this protein is getting activated. That can tell us what part of learning this protein is involved in. So to really understand this signaling, we pioneered in this technique of developing biosensors. These are Fred biosensors where, um, yeah, just a couple of minutes. So these are Fred biosensors where we have, once the proteins are activated, they undergo some conformational change, and that we can detect if we have a donor fluorophore and an acceptor fluorophore attached to it. And this is where I did all Perny sir's molecular cloning classes came into handy, where what we do here is just simple molecular cloning to bind or to attach a particular subunit, protein subunit. In this case, it's CAMK2 protein. We can bind it to a donor fluorophore, which is GFP most of the times, and an acceptor fluorophore, which is ready to accept the donated photons. In this case, this is dim Venus. Now, using these sensors, this is pretty simple design. We can then study protein activity with really high spatiotemporal resolution. And I can give you two examples. Now, both the proteins are shown to be important for learning, but both of their activities are completely different. If you see, you press. In this case, protein gets activated only in the synapse. So now this is a sensor that is genetically encoded into the cells with a specific promoter. And you see that this activity only gets activated after the activation. So they get activated in a synapse, and then you have an increase in the spine volume. Whereas, let's see, if you look at a row A activity, you see that the activity of that is a lot more spreading. So can we play that again? Sorry. So if you see, if you induce plasticity here, it will spread throughout the dendrite and will not be restricted here. Anyway, so it's not playing. Okay, it's fine. So with these sensors, we can get a lot more quantitative information. So we use these sensors to ask a plasticity question. So we optimize the CAMK2 activity and we measure the lifetime. And so as compared to the synapse-specific role that I showed in the past, we found that a lot more delayed and global CAMK2 activity underlies this form of plasticity. So you'll see that it will be activated throughout the dendritic shaft, and then you'll see the changes. And we combine that with electrophysiology to see that there is actually an increase in the EPSP amplitude. Sorry, in the interest of time, I'll just move. But what we found is that using these novel techniques, we can now get to more quantitative understanding of how these proteins are getting activated during learning and can actually link a specific molecular signaling during a specific form of learning, which is what we are trying to do in the lab. And then, of course, we can use these sensors to now see during the learning, when the learning is disrupted in any kind of disorders, what happens and how do these specific protein signaling get affected. So yeah, I'll just skip because these are all details in telling us how does that CAMK2 get activated. It's a calcium activating sensor. But yeah, I'll just skip that. But yeah, thanks. And I'll be happy to have questions. I don't think we can have questions now, but I'm happy to talk to you guys later.
Yeah, sorry, we'll not have questions at the end of the awardees talk, but we encourage the participants and faculty to interact with them offline. And I hand over to Kritika to continue the program. Thank you.
Thank you, Dr. Anand Jain, for your insightful talk. I now welcome Dr. Devi to introduce the next awardee for the session.
Dr. Devivasha Bordolai, a proud alumni, is an assistant professor in the Department of Biological Sciences at the Indian Institute of Science Education and Research, ISL Bhopal. She earned a bachelor's degree from our department, and then she pursued a PhD in cancer biology at the Department of Biosciences and Bioengineering, Indian Institute of Technology, Guwahati, India. Following a PhD, she moved to USA for postdoctoral research, joining Professor David B. Wiener's team at the Wistar Institute, Pennsylvania. a globally renowned research group specializing in nucleic acid vaccines and immunotherapies. Her current research focuses on developing novel immunotherapy approaches, including monoclonal antibodies, bispecific, multispecific immune cell engagers for targeted cancer therapy. She has filed 6 US patents so far and has over 70 publications, including original research articles, reviews, books, and book chapters. Her research grant has garnered 5,000 citations with an H index exceeding 30. Her scientific contributions have been widely recognized through prestigious accolades, including selection for the DBT Ramalingasamy Award in 2024, the Young Scientist Award, the Vista Science Accelerator Award of 50,000 US dollars, providing funding to support innovative technology-driven research programs, along with invitations for presentations at prestigious meetings, such as the plenary talk at the annual meeting of the American Chemical Society, a talk at the Special Ovarian Cancer Conference by the American Association for Cancer Research, invited talk at Gordon Research Conference on antibody biology and engineering. While her work has also featured a special editorial highlight in esteemed journals like the Journal of Clinical Investigation and the Journal for Immunotherapy of Cancer. Wishing you future success in future. Now I invite Dr. Devi Varsha to deliver a lecture.
Um, Hello, everyone. Very good afternoon, everybody. Thanks a lot for such a warm introduction. And as Dr. Anand said, it's really a very gratifying moment for us to be back at the place where we were students a decade before. So thank you, and thanks to this conference organizers for having me here, and also honoring me with this wonderful award, which I will always cherish. So thank you so much. So without much delay, I'll just start with my work. So switching the gear a little bit from basic science to translational, what I work on is I develop novel bio, and some of my recent work focuses on developing novel biologics for targeted immunotherapy of ovarian cancer. So ovarian cancer, we all know. I don't think there is any specific introduction needed to this panel. Basically, ovarian cancer is bad. So this is the cancer of the ovary. So the cancer of the ovary and the distal fallopian tube, which connects the ovary to the uterus. So this whole thing comes under ovarian cancer. This is the deadliest gynecologic malignancy. There is very high incidence rate of ovarian cancer if you compare to other gynecologic cancers. And it has very low survival rate, especially at the advanced stage, like many other cancers. But one of the major problem with ovarian cancer is that it is diagnosed often at very advanced stage. So that's why it is also known as silent killer, where the survival rate is very low as 15 to 17%, and that is why it is also known as silent killer. It is mainly because of the very not so distinguishable characteristics or symptoms exhibited by the patients. This is a global scenario, but the scenario of India is no different as well. So this is one of the most predominant cancer in female in India as well. And if we see the treatment modalities which are available for ovarian cancer, so if we talk about immunotherapy, which has greatly revolutionized therapy for many cancer, in that specific mention has to be of checkpoint inhibitors. But for ovarian cancer, checkpoint inhibitors has not been very successful. In fact, the response rate, as per many trials, it is only 10 to 20%, which is not as encouraging as some of the other cancers like lung cancer or melanoma. So this definitely emphasizes to identify novel targets which not only modulate the tumor cells but also the tumor microenvironment. So this one target that we have come across, FSHR, so we thought this can be a very good target, follicle stimulating hormone receptor. So this is basically involved in reproduction and fertility related function. And why we think this can be a good target? Because FSHR has very selective basal level of expression on normal ovarian cells and ovarian endothelium. But it has around 50 to 70% expression on ovarian cancer cells, and it has high expression on chemo and aggressive ovarian cancer subtypes as well. And when we tried to analyze FSHR expression on 48 different healthy human tissues, with a comparative analysis to ERBB2 or HER2, surrounding which many molecules have already been developed. They are, and several others are in clinic. So FSHR has barely any detectable level of expression in all these healthy human tissues, which definitely indicates its safety profile. So as a translational lab, actually, we have first tried to develop antibody against FSHR. And our aim was to develop antibodies which bind to a conformational epitope of FSHR. So we have designed FSHR vaccine, and then we have immunized mice. And then we have developed antibodies, but cutting screen our clones using this FSHR expressing cells as well as FSHR negative leukemia cells. Well, we had several hits, so a diagonal pattern indicates a very good clone having good specificity. So although we had several hits, but this clone D2AP11 was the most specific and had a very good binding. So we have further characterized this clone. So these are all the binding data on different ovarian cancer cells. And as you can see from the shift in the peak, there is very good binding to different ovarian cancer cells which are having expression of FSHR. And then for a very good antibody to develop, it is very important that the antibody does not have cross-reactivity to our vital healthy human tissues, right? Otherwise, it will have adverse reactions. So that's why we actually tested this antibody's binding to several vital healthy human tissues like pancreas, lung, heart, small intestine, colon, uterus. And there was no significant binding. There was, of course, some binding to ovarian, fallopian tube, endothelium. Because as I said, they have some basal level of expression in them. But when we have checked them on ovarian cancer cells, different ovarian cancer tissues of different pathological conditions, as you can see here, the IHC staining, there is strong binding in all those ovarian cancer tissues. And this antibody was able to induce ADCC, antibody-dependent cellular cytotoxicity, with an EC50 of 202 nanomolar, which is great, but not the greatest. So now we thought how to improve on the potential of this antibody. And for this, we have designed this bispecific T-cell engager. So as the name suggests, bispecific, so it has bidirectional specificity. So this canonical bispecific T-cell engagers, they're designed by connecting the single chain variable fragments. So if you see an antibody, the tip part of two antibodies, they're connected with the help of a flexible linker. And now this is like one molecule which works MHC independently, and basically it bridges the gap between the immune cell and the tumor cell. So it establishes a bridge between both cells. So from one side, it will bind to the tumor cell, and from the other side, it will bind to the immune cell. And this activated immune cell now release perforins, granzymes, and other cytotoxic molecules, which will cause the targeted destruction of the cancer cell. This is a very emerging approach with 5 FDA approvals so far, and mostly in the last 2 years, which definitely emphasizes the need of this kind of research in our country as well. And also in US, it's a very new field. So, so we have designed an FSHR targeted bispecific T-cell engager using our FSHR clone, and that we have developed as a good antibody. And then the first thing was, of course, to check its expression. And there was, when we confirmed its expression along with relevant controls, next thing we wanted to see whether it has now bidirectional binding, which is binding to FSHR and the other side to T cells. So this is binding to FSHR expressing cells, be it natively FSHR expressing ovarian cancer cells or FSHR transduced ovarian cancer cells. We can see a very nice shift in the peak indicating its binding. And then we have determined the CD3 binding, which we analyzed on primary human T cells. So now we know that it is binding from both the sides, from one side to FSHR and from the other side to CD3. So now the next important thing is to know the functionality, whether it is doing killing of the ovarian cancer cells or not in presence of immune cells. This we have done with Excelligence real-time cell analysis assay. And if you see here, there was so this red line is no antibody treatment, And this blue line is treatment with this bispecific T cell engager. And in both the cases, we have used immune cells. So we have seen this D2AP11 TCE was very significantly killing these ovarian cancer cells with an EC50 of 294 picomolar. And I just want to show a small video. So these green cells are cancer cells, and these small cells, what you are seeing are human PBMCs. So this is the treatment group. Here you can see in presence of this bispecific T cell engager, these small immune cells are surrounding around these cancer cells and they're killing the cancer cells. And at the end of the experiment, we barely see any live ovarian cancer cells here. Whereas in the control, even in the presence of immune cells, the cancer cells are growing happy, healthy, and we can see a well confluent with cancer cells. Since ovarian cancer, as you know, it has many cancer driver mutations, and also subsequently, they're resistant to a lot of different drug targets, like there's like BRCA1, BRCA2 mutation, and subsequently they are exhibit resistance against PARP inhibitors. So we wanted to see where our biologic best fits in. So that's why we have chosen a panel of different human ovarian lines with all these different mutations and also different drug resistances. And it was very encouraging to see this, that the biologic that we developed was efficiently killing all these different ovarian tumor lines. And then we have observed its in vivo efficacy in a humanized ovarian cancer challenged mice model. And also for this, the route of delivery was direct DNA, so that we can limit the repeated dosing, which we often have a problem in case of protein. So we have delivered in the drug in the form of DNA followed by electroporation. And then we have monitored tumor growth over a period of time in this humanized ovarian cancer challenged mice model. And as you can see here, there was significant shrinkage of the tumor burden. And this result was also very encouraging. But then next we thought, why to engage only T cells? Why not to engage additional immune components? For example, NK cells. Because most of these cancer cases, they have very less infiltrated immune cells in the tumor microenvironment. So if you can bring in more immune cells, then it will always be providing additional tool. So that's why we thought of engaging NK cells, and NK actually offers a lot of different advantages. It has high killing capacity. It has lesser cytokine release because of what immune-related adverse events, which is often a problem in case of T-cell therapies, like, for example, CAR-T therapies might not be that big of a problem for NK-based therapies. So for targeting NK, we have chosen this marker called Siglec-7. So Siglec-7 is a pan-NK marker, and this is also a recently identified glycoimmune checkpoint. So how this works actually, so this Siglec-7, it is present on the surface of NK cells, and its ligands, which are sialic acid, they are present on the surface of target cells, in this case, ovarian cancer cells. And basically, this interaction of the ligand and receptor leads to the activation of a pathway which suppresses the immune cells. And because of that, the immune cell will not be able to mediate cancer killing. So we thought if we make an antibody specifically against Siglec-7, then this will be able to block this interaction, and as a result, immune suppression will not be happening. So we have actually developed an antibody against human Siglec-7, and these are fully human antibodies because we have developed them in humanized mice, but I'm not going into the detail of it in the interest of time. But cutting the long story short, we are able to have some good antibody clones against Siglec-7, which had very good binding to human NK cells. As you can see here, over 90% binding to human NK cells. And this binding was to both cytotoxic as well as helper NK cells. And then when we have tested these antibodies for killing on ovarian cancer cells, as you can see from the graph as well as from the images, when this antibody was added to the ovarian cancer cells, OVCAR10 cells here, in the presence of either human PBMCs or purified human NK cells, there was significant killing. So this is the control. You can see cells are growing, and it's a fully confluent well, but here cells are almost dead. And this is post like 48 hours treatment. And then, of course, the in vivo study. So here we have again given a DNA delivered anti-Siglec-7 antibody, and one dose of this DNA delivered anti-Siglec-7 antibody was very significant in significantly attenuating the tumor burden as well as enhancing the median survival of the mice significantly. And then we thought, why not to now design a bispecific NK cell engager? So we replaced this CD3 arm now with anti-Siglec-7 arm. So other arm we kept same, that is anti-FSHR, the D2AP and anti-FSHR antibody we developed. And the other side we put this anti-Siglec-7. So now this one molecule, which we call it is call as DB7.2 D2AP11 NK cell engager, NKCE. So this molecule we expect it to bind to NK cells as well as FSHR positive ovarian cancer cells simultaneously. So after checking its expression the same way, we checked its bidirectional binding, in this case the binding to FSHR, and then Siglec-7, which is predominantly present on human NK cells. So we have checked the binding by flow as well as immunofluorescence along with relevant positive and negative controls. And then, of course, the next important thing was to see functionality. And for this, actually, first we checked on a panel of different non-FSHR expressing cells, because we don't want this biologic just to go and target anywhere. We just want this biologic to go and kill only those cells which are positive for FSHR expression. So that's why so my postdoc boss always says it's the negatives which always makes your positive stronger. So actually we have included a lot of negatives. So AGS, that's a human gastric adenocarcinoma cell line. So this does not have expression of FSHR. So when we have tested on this cell, so we have added effector cells, which are human PBMCs, and then also we have added this bispecific NK cell engager to it. And then when we have checked both as well as graph in the images, you can see there was no killing even in the presence of this bispecific NK cell engager, which indicates that this is highly specific and it is not just going and killing anywhere. But contrary to that, when we checked on a panel of different human ovarian cancer cells, which are having different expression of FSHR, for example, CAOV3 cells here, so we can see this bispecific NK cell engager in the presence of human effector cells, either PBMCs or purified NK cells. There was significant killing in these cell lines. And as you can see also in the images, in the control where cells are growing only in the presence of immune cells, but when you add this NK cell engager, there is significant killing. And this killing was actually, so we have verified this in multiple lines. And this was actually achieved at an AC50 of 142 picomolar, which is actually very impressive and very comparable to some of the approved bispecifics. And then, as I said, we have checked in like a lot of different negative cell lines which are not having expression of FSHR. And as expected or as desired, we did not get any killing in those. But when we checked in different FSHR expressing ovarian cancer cells, there was significant killing in those. I'm not showing all the data, of course, in the interest of time. And this is the in vivo result. So here again, we have developed an ovarian cancer challenged humanized mice model because we wanted to give human PBMCs, and also the biologics that we are developing are all human biologics. So we cannot do in an immune competent mice. So we have used these nude mice, and then we have developed this as a humanized ovarian cancer challenge mice model. And our delivery in vivo was again DNA here. The reason for using DNA is that DNA is highly stable, and you inject the DNA in the muscle, and then it will stay there, and muscle will be acting as a factory of this protein. So basically, it helps reducing the repeated dose, because proteins are often having a shorter half-life. So this DNA delivered a bispecific NK cell engager, 2 doses we have given, but there was significant shrinkage of the tumor burden, as well as enhancement in the median survival of mice, almost by double, which is definitely very encouraging to us. And also, this is the first description of a Siglec-7 based or any member of Siglec family based NK cell engager, whose function we have shown, and also we have demonstrated it as an important tool as well as approach. And we have shown its efficacy in vitro, in vivo against human ovarian tumor or any other human tumor. Of course, if we have to summarize these few studies, so we have developed multiple new tools for ovarian cancer immunotherapy. Antibodies which bind to the conformational epitope of FSHR, because we have screened all the antibodies on a cell, not by immobilizing it on the surface. And then we have developed FSHR targeted T cell engager with high potency in vitro and in vivo. Number C, we have developed antibodies, fully human antibodies against Siglec-7, which also activate as also work as CPI. Its function is very reminiscent of PD-PDL1. This is an NK cell checkpoint. And then we have utilized that anti-Siglec-7 antibody to develop a novel bispecific NK cell engager. And also, if we notice, when we have used the anti-FSHR antibody for killing in those ovarian cancer cells, it had a potency at nanomolar level. But when we have made that in the form of bispecific T cell engager or NK cell engager, we were able to bring up the potency 1,000-fold higher. So basically, this demonstrates a novel approach of FSHR targeted T cell engager, as well as a very novel approach engaging Siglec-7 or any member of Siglec family to develop a novel NK cell engager and its efficacy against human cancer. What we have shown, but it has definitely can have applicability for other diseases of concern as well. So these are some of the byproducts, of course, some papers and journal highlights. But also happy to share that this FSHR targeted T cell biologics and antibody, they are currently under license agreement processing for further translational development. With this, I would like to conclude my speech. Thanks to my collaborators on this work, David Weiner, then I was working at Vistar, UPenn, Michael Betts, Carl June, and some of the funding agencies. And currently, I'm an assistant professor at ISR Bhopal, but I'm just setting up my lab, pretty new. And of course, my alma maters, without who I would not have been able to talk here today, IIT Guwahati and SRM. I just have to say that like I started my research work first at SRM itself. So I was doing a project with Usha ma'am on plant. And then I got a Best Undergraduate Research Award. And I was very happy. And I just thought, okay, this is something I should do actually. Because I just used to try like, oh, I should top this semester. But somehow I failed. And then when I got that award, it was like very encouraging moment for me. And I thought, okay, maybe research is something I should pursue. So sometimes like small incidents make bigger impacts on your life. And I always write that in my CV, like, oh, I got the Best Undergraduate Research Award. So it has really a very great impact on my life. Usha ma'am, she has been a great mentor. And I feel if you have such mentors, everybody can do a lot of great things. So thank you so much. Okay, sorry. Yeah, thank you. Thank you so much.
Thank you so much for a thought-provoking session. Now I invite Dr. Manoj to introduce the next awardee.
Good afternoon, everyone. It's my absolute pleasure to be here today to introduce someone whose vision, determination, and entrepreneurial spirit have made a significant impact in the field of genetic engineering and biotechnology. Mr. Sujit Kumar Singh, the visionary founder of Cytogene Research and Development, who is being honored with Young Entrepreneur Award today morning. Mr. Sujit Kumar Singh is our alumni of 2010 batch, completed his BTech Genetic Engineering. Then he completed his MTech Biotechnology in 2019 from Rama University, Kanpur, and secured his executive MBA degree at IIM, Jammu, during the year 2022. And about the Cytogene Research and Development, it is a contract research organization dedicated to offering research services in the field of life science and biotechnology. The organization specializes in the contract research in collaboration with various research and academic institutions, independent testing services of the samples of various researchers, training and awareness programs on sophisticated instruments. Under Sujit's leadership, the Cytogene has grown into trusted name, empowering thousands of research professionals through end-to-end sample analysis services throughout the India. As a graduate of our institution, Mr. Sujit Kumar Singh has consistently demonstrated not just intelligence and ambition, but also a commitment to giving back to the community. He embodies the value we strive to instill in our students: creativity, perseverance, and the belief that with work hard, anything is possible. The Young Entrepreneur Award is not just about the business success. It is about the vision to create something new, the courage to take risks, and integrity to build something meaningful. Mr. Sujit Kumar Singh has certainly proven that he possesses all of these qualities and more. I welcome Mr. Sujit Kumar Singh to deliver the speech on his success story behind his achievements. Congratulations, Sujit. The stage is yours. Thank you.
Thank you, sir. Am I audible? Yes, good afternoon, everyone. First of all, thank you. Thanks a lot, all the organizing team, our beloved teacher. I'm really feeling pleasure to, again, speaking something in front of our teacher. It was way back when I used to listen them, and now it's my turn. Fortunately, unfortunately, this was like, I'm lucky, I feel I have been here for 4 years during my BTech. And the first turnout, like when I used to go people, when I started this lab, they used to ask like, who is owner of this cytosine? And at that time, I was only 23, 24-year-old. So I cannot say it is me, otherwise they won't believe. At that time, there was no startup, no stand-up. So I used to say, Sujit sir is there who is owner. So they give their like card and ask Sujit sir to talk to me. Usually I come back to home and then I call and talk to them. Still there are, I think, at least 100 of people, they do not know my age still, but they are in touch with like from last 10 years or more than that. First lecture I delivered was in Graphic Era University, Dehradun in 2013. At that time, people usually talk about only research and all. They don't talk about any business or entrepreneurship. So they are not at all interested to listen about business. They are asking if you have anything to research, you can represent. So I presented DNA barcoding. What I have done project here in SRM under Rex sir and guidance of Parni sir. So from there, I got a platform with DNA barcoding. I have done at least 30 to 40 lectures on DNA barcoding only. But that is just a platform. I have to promote my own business, but if I'll talk about business, they won't give the platform. So I have to use that DNA barcoding topic just to promote ourselves. So here this PowerPoint, it is nothing but just a demo because I have to submit something before 5 o'clock yesterday, so I sent something. So nothing much is there. I'll talk about more or less, I'll talk about money. From morning only, I've seen there are a lot of topic and research work. Like somebody is talking about cancer, somebody is talking about Y chromosome, somebody is talking about diabetes. How life goes with money. Okay, all the talk you have like listen from the morning and tomorrow evening, all the talk if you listen, you will find it is funded by DBT, Government of India, ICAR, ICMR. You will not find any business or local, like corporate industries are funding any research. And that is where we are lacking. In the morning, Mohan sir told, like, we are very behind. Niranjana sir told, we are very behind in terms of research. Why? Because in India, we do not care about research as a private organization. More or less, if you'll find 95% all the research are based on government fund. So if government will not give fund, no research will happen. So that's what I have taken a different path. Usually you people do BTech and then MSR MTech in US, Canada, you move there. I moved back. I went to Lucknow after studying BTech and I'm there only. So usually in Lucknow, people speak in English only 2 times, either during interview or when they are on high. Okay, so pardon me for English. I'll start. What you have to actually think. My friend Anand Jain has told a lot about memory and what we remember, what to forget. I'll just give a brief. Like the Guardian has published one paper and he told like usually people talks 15,000 words, especially girls. Ladies talks 15,000 words in a day. Gents talk around 5,000 words a day. Working lady and gents. And almost they are making 150 to 250 promises per day. How many we remember? Hardly 4 or 5 or 10. So obviously promises are made to broken. So that's how we will go. Now, first of all, I'll introduce about cytogene. Now I don't need any research work to introduce about cytogene. So fast forward, I'll go with this journey, and then we will come what actually you have to think and what you have to do. Cytogene, as already Manoj sir introduces, like it's a research organization, we do in collaboration with various universities, colleges, and everything. We are standing here in a premium institute. Please remember, you are in a premium institute. You are having all the facilities here, but you won't believe we usually and regularly get a student who has completed their MSc in biotechnology and they are first time watching micropipette in our lab. Can you believe? A lot of gathering is there. That's what diabetes, Mohan sir has mentioned, no? Gathering is more. Our efforts are very less. He has shown one example also, like 9-year-old girl. She has saved almost 2,50,000 rupees, right? During her tenure. Only 2,000 is cost for one test. That disease is very rare. If I'll test 1,000 people, maybe 1 or 2 people will get positive. So for 1,000 people, how much amount is wasted? 20 lakhs. So everyone can't go for that testing. That is the major problem. But sir has told like they are offering free also, and they have standardized many things during this period of work. So that's what I used to say, like if you are doing any research work, just keep in mind your H index will be increased, impact factor will be good. But what about others? You cannot left everyone just because of me. I am surviving more or less. That is fine. No. Established to bridge gap between academic and research institution in 2011. 10 I pass out and immediately I moved on. In 11 I was started it. This is now we have recognized with many other things. Mission to revolutionize biotechnology research through innovation, collaboration, and excellence. Just an example I'll give. Like in out area, there is very difficult to get GCMS services. I hope you all know GCMS, right? When we started this in 2018, we were offering it at 3,000 per sample. Then after 1 or 2 years, we have standardized this to 2,000 per sample. And then we have standardized to 1,800 per sample. Now we are about to offer at 1,500. Our target is to go to 900, below 1,000. So people who are about to give their sample, one sample at 3,000, they can give 3 samples at the same cost. So that's what we are targeted to do. Of course, everyone has a vision to grow their own company in a world-class level. Okay, from where the concept came to initiate a lab? Obviously, you are studying in classroom. So if somebody is showing attitude, you must have thought, no, I'll run a company and I'll hire you as a secretary. So usually people come and initially they like come with this beginning. I had given many like interviews, but usually people says you are only undergraduate, we cannot hire you. At least postgraduate you need to do. I was in ISC Bangalore. So I thought they are offering, still if you'll see, they are offering undergraduate as a PhD student. So I went there, again they say this is like same thing. You have to complete your MTech RMS, then you have to come back. So then I asked like where I can do research. I was frustrated. So they said, okay, go open your own lab and you do your research. So from there it came in my mind, okay, I'll open my own and I'll do. So that's what I'm doing from there. Okay, building foundation, company was made in 2011, starting with a small research lab. Initially, we are doing only training for the student who have not seen any instrument and all, because in that, I have studied in SRM, so I was in a very top with them. And then one thing is very important, funding. People usually seek funding. I'll suggest you do not use Okay, suppose if you are having 5 lakhs in your hand and your budget is 20 lakhs. So what you do, 5 lakhs you arrange from you, your family and your friends, and 10 lakhs you take a loan. That is wrong decision. You try some business which actually need only 5 lakhs. And your family, friends, 5 lakh will be there to run your business smoothly. Because after establishment, you need almost equal amount to continue the same business. So I'll not prefer at all for any loan or something, because if you take a loan, you will have a target to earn. So initially for 1 year, it will go smooth, and then they will be pitching every time. You have to do wrong things, manipulate the things, because you have a target to do. So initially foundation, we have collaborated with various academic and all. Growth and expansion, whenever we do like what we had done initially in 2013 or 14, we have shifted our lab and we have established a complete state-of-art lab to work with modern biotechnology like PCR and all, everything we had at that time. And we started with the training along with the PhD training also. Major challenges in our field, I feel, I already told you, this is a premium institute. We cannot compare low awareness among the researchers. There are many PhD students also who do not know how to type in MS Word. Will you still believe? Yes, we are facing every day. There are thousands of students, they are doing their thesis, but they do not know how to type in MS Word. MS Excel is something different at their level. They think like they need coding for MS Excel. So that's how people are having. So we have to train them on computer also, not only like electrophoresis and all, for computer also, understanding the exact need in where they are having a problem. That is the main need of this business. And that's where we have grown throughout, because I have almost 14 years of experience. So like doctor, I used to see what he need. So immediately I jumped to a conclusion, like this is what actually they are needing. Building a skilled team, again a major task. You will hire somebody, you will train them with 3 months, 6 months, and then they will leave. So again you will hire, you will train them, they will leave. Before 4 or 5 years, I came here for a talk. Same problem I discussed with Parni sir. Sir, people are usually come, they learn and they leave. Sir told, don't worry, in SRM also they come, they leave. So your lab is very small. Why you are worried? Just hire and fire. So from there we triggered it. I'll keep hiring. If they will leave, no problem. Again I'll hire. So every time it is managed. So many things I have seen, as I have already mentioned, Parni sir has continuously guiding me. Earning trust, again the main, if you are doing any business, you need a trust. Because on business is totally based on trust, either based on money. So every time we cannot keep money in our hand to earn that. Struggling with the deadline of report, that is major problem now also, and almost everywhere. I think even in SRM also, sometimes you will feel like if you have sent sample somewhere, they have given deadline of 1 week, it is almost 15 days, 30 days it passed, then you are getting results. So that is a common problem within all over India, I think, not only in our. Overcoming obstacle, easy reach online, offline. We have offered actually online classes also for those who do not understand the technical parts. From there, we have got a good gathering. Strong reference pattern, like we do not start any work until they are giving any reference, because we have to answer how we performed it. And then extensive training program for the team, installing feedback system. This is what actually I learned a lot. When we discuss with the people like what work they have got, it is good or bad, they actually don't know what they have got. Then we realized, okay, they got GCMS report, but they don't know what to see in this report. So we have started post-delivery, after delivery, we have started a training program also. Like whatever result we have given you, how you will see in that result. What are all the major points you have to see? So there, again, it was triggered for our business awareness towards fraudulent activity in the market. There are many players who took the sample just because of name, like I'm going to give cytogen and nobody knows where they went. Have you seen this? If somebody is from Mumbai, they must be knowing Dabbawala. Their accuracy is 6 sigma. So continuous improvement, always strive to improve. That is the only basic you have to do. If you are planning for any business or anything, you have to do. What I have done, almost 3,500 plus researchers and students, we have done training in our laboratory, published more than 90 plus research. This is not my only whole lab. And then partner with 250 universities and industries, including like IIT Kanpur and all. And IT Trichy is also one of our partner, and IT Raipur and all. Recently, Cytogen has completed contract research project in State Forensic Lab in Chhattisgarh. It is in Jagdalpur, next light area. Usually they are getting dead body in the jungle after any like encounters. So they are isolating mosquitoes and flight from the dead body, and they are like doing research on that. So some part they are sending to us for the analysis and all. Cytogen has completed research work. This was actually interesting. That was already a completed project, so I can explain. Like in a plant, you might have seen there are some holes or any parts where water will be deposited, and because of that water, there is a microflora developed. So they are studying like in that particular part in the plant how everything is changed, like ammonia, heavy metals, and many things they have searched. So in Indian location, we got that project to complete. Currently, we are having many ICR lab, mostly beans, like different type of beans ICR lab are developing, though they are sending us for nutritional value and all the things. Impact through workshop, yes, this is one of the game changer, because initially I have started in DNA barcoding, so I keep it till now. I am doing this. Most of all people in Lucknow, they know like if you have to see something in DNA barcoding, Sujit is the right person to discuss. So still they are coming with us to discuss. And apart from that, we have organized like more than 70 outdoor workshops, and including those colleges which they are not having even basic facilities, like they do not have light. So they call for like generator and all everything. So sometimes we have to face, but yes, it was a nice experience with those person. They do not have micropipette, they do not have freeze or incubator. So we used to go there with all the instruments, and we did training almost 5,500 students. During COVID, we have organized almost 65 plus online workshop, pan-India. Mostly professors, like senior professors were joined who had studied at their time in botany and zoology. But now they want to learn like molecular biology, genetic engineering, and mainly bioinformatics. Like we have last year, we have organized one international workshop also. Social impact, what we do, Cytogen has demonstrated a strong commitment to. These are all the things going on. Timeline, like how year by year we have gone through. 11 we have established, 12 we have established biochemistry and immunology, 13 in workshop we have started, 14 we have organized first international workshop, then 15 training program to in-house training program for graduates and researchers, 16 contract research services we have started, and then we have gone through like all the approvals and improvement. What we need to do, establish international collaboration, like 2 universities we have already collaborated for research work and project with our laboratory, and we are planning at least 10 by this year, by the end of 2025, and improve further in the field of biotechnology, addressing most major challenges. What I will suggest to you, be problem solving. If you are having any solution for any problem, your business will never die. Just imagine if you are having any issues, you are giving solution, it will never die. Your business will always sustain. Never stop learning. Always have to strive to learn. Build a strong network and collaboration. Obviously, IIM and IIT do. Adopt innovation. Strategy is one of the major part in business if you are planning, other than your research. Great strategy has pieces, but they form a coherent whole. we usually have a thought or idea. Can you measure it? If any thought comes to your mind and you can measure, that is idea. If you cannot measure, that is not idea. So sometimes we used to think like whether this idea will succeed. First you think whether this is idea or thought. Thought is different from idea. If you can measure your thought, that is idea. Yes, it can be succeeded if you are going in a well manner, established manner. So usually we are having many thoughts, but we do not go seriously for that. Okay, that's all. My time is over, I think. Thanks a lot.
Thank you, sir. We next go forward with the plenary session 2, theme Genomics and Computational Biology. This session is chaired by Dr. M. Parni, and I invite him to introduce our first speaker for the day.
I'm happy to introduce Dr. Muthamazharan. Dr. Muthamazharan earned his postgraduate degree in biotechnology from Madras Kamaraj University, Madurai, and completed his PhD in plant biotechnology from the National Institute of Plant Genome Research, New Delhi. His doctoral work focused on developing genetic and genomic resources in foxtail millet. Currently serving at the University of Hyderabad, Dr. Muthamazharan leads a research team investigating pre-harvest and post-harvest traits in foxtail millet. His team extensively employs next-generation sequencing approaches to decode genes and study their role in stress responses. Dr. Muthamazharan's outstanding contributions have been recognized through national and international awards, including DBT Innovative Young Biotechnology Fellowship, INSA Young Scientist Medal, and Fulbright Nehru Doctoral Research Fellowship. In 2024, he was selected as a INSA Associate Fellow and is also member of the Indian National Young Academy of Science. Dr. Muthamazharan work continue to make significant strides in millet genomics, bridging the gap between fundamental research and its application in sustainable agriculture. With this, I invite Dr. Muthamazharan to deliver the lecture.
So yeah, good afternoon, everyone. After listening to all different topics, now you are going to listen about plants, right? So after hearing that you are from UG and PG, I have changed my slides a little bit to make it more simpler. And always the problem is talking to an audience after lunch. So I don't want to sing a lullaby to make you sleep. Right? Okay. So hope you all would have heard about millets. Yes or no? Isn't it? Yeah. And we celebrated the year 2023 as the International Year of Millets. And I did my PhD with millets, and I still continue working on millets, and that is why my lab is called the Millet Lab. Right. I can say proudly that among all the central universities, the only lab that work exclusively on millets is my lab at the University of Hyderabad. So I hope you all know that from Kashmir to Kanyakumari we cultivate at least one variety of millet in every state. Millet is uniformly cultivated in almost all the states, right? And I work on a crop called foxtail. Actually, that inflorescence looks like a tail of a fox, so they call it as foxtail. And we recently started working on other millets also, right? Foxtail millet, it is a diploid genome. It's a very small genome. It's similar to rice. In fact, rice has 12 chromosomes, whereas foxtail has only 9 chromosomes, right? And its life cycle is almost similar to that of rice, and it has a very good, genetic diversity. We have different. You can see the diversity, at least with respect to the panicles here, right? The inspiration came from the literature. When we see the literature, we could see that two millets are invariably mentioned, particularly in Tamil literature. One is foxtail, the other one is kodo, varagu, and thinai. Right? I hope the others people from other states cannot read. Right? It is seen that before rice, our ancestors were eating not just millets, different other diverse diets, among which millets occupied a significant proportion. If that is the case, then when did rice came into picture? That question was answered by a very recent study published in Nature Plants in 2020, which says that the rice which we consume now, Varisa sativa indica, was domesticated somewhere in the northern part of UP approximately 4,000 years ago. So before 4,000 years ago, maybe we could have been eating, our ancestors would have been eating millets plus other species which slowly vanished. In fact, it fastly vanished after Green Revolution. The same was established by another Indian study published by Birbal Sahni Institute of Paleobotany. They say it is little more before. Rice was domesticated in India around 80,000 years ago. So it's understood that before the advent of rice, maybe we would be consuming millets, and that is where we are seeing a lot of mentions about millets in the literature. Now, why it is not moving? Right, okay. So we chose that crop to work on. The problem is that before 2012, no much work has been done on this crop. In fact, whenever we talk about millets, people used to say, don't work on millets. Right, that time nobody knew millet much, and it was unwelcomed. They used to say, you better do that work in rice, you better do that work in wheat, or at the max do it on tomato. Don't touch millets. But fortunately, when I started my PhD, its genome was sequenced by a Chinese group as well as US group. We had no role in that, but we took that information, whatever the genome they have sequenced, and we started working on it. Right. I have shown you the genetic diversity of the millets. We have very good genetic resources available for millets with NBPGR and as well as ICRISAT. Now, for any breeder to go for a crop improvement, they require genomic resources. So we wanted to use the genome sequence information to venture into as much as things as possible. So today I'm going to show you what are all the things that we did with the foxtail millet genome sequence. Right. The first comes is marker discovery. Molecular markers are very much important for breeding. Any breeder, if you know, India didn't permit GMOs. Except BT cotton, we don't have any GMO. So all the crops that we are consuming are the products of breeding. So if they have to do breeding, they have to have molecular markers. They have to link those markers with the trait, and they have to do breeding. So our first objective was to develop molecular markers. I hope many of you will feel it little dry So I'm not going into the details of it. What we did is we took the genome sequence information as well as the transcriptome sequence information. Then we tried developing different types of markers. I'm stopping there only, right? Then you have to associate those markers with the trait. You should show this trait or this marker is linked to this trait so that the breeder can use those markers for marker assisted selection. So for that, we attempted association mapping. Even in the morning, you would have listened to the word GWAS, right? I hope many of you would be knowing the GWAS pipeline, right? So these are using SSR marker. I'm not going into that detail. So when NGS has become cheaper, we were able to resequence as much as accessions that we had with us. We had approximately 184 foxtail millet accessions. We resequenced all the 184 accessions to identify a good number of SNPs. Those SNPs were used to perform genome-wide association study. When you perform GWAS, you will get such a graph called Manhattan graph or Manhattan plot, right? So this shows the different SNPs distributed across the genome, and it's if you see if an SNP is lying above this threshold level, that means that SNP is strongly associated with your trait, right? So by this way, we were able to identify several genes. Unfortunately, many of the genes were of unknown function. Now we are trying to characterize them to identify what they actually encode for. Millets are also known for its nutrition. Right? You would have read also. Right? People always talk good about millets, particularly for its nutritional value. Millets are particularly rich in micronutrients. If you see the amount of calcium that ragi has, where is ragi? Finger? Is it not a finger? Right. The amount of calcium that finger millet has, it is 100 gram, right, is equivalent to 3 glasses of milk. So millets are at least 5 to 6 fold nutritionally superior to the so-called rice and wheat. But if we consume millet, are we going to get all the micronutrients that the grain has? The answer is maybe not, because it has a particular antinutrient which chelates these micronutrients. We call that antinutrient as phytic acid. Millets are also rich in phytic acid content, right? So we wanted to identify which are all the genes linked to this phytic acid trait. So we performed GWAS. What we did, we cultivated around 126 foxtail millet accessions in 2 locations. Then we performed resequencing, and then we identified the SNP markers. With those SNP markers, we performed GWAS. Then we identified a marker, a SNP, lying on non-specific serine threonine protein kinase gene is tightly linked to phytic acid content. Now one of my PhD student, who is also a PMRF fellow, she has knocked out this gene, and those knockout lines are low in phytic acid content. I'm not showing that data, but using this SNP, we developed a PCR-based marker which can easily distinguish low phytic acid content lines from high phytic acid content line. Now, if you are given a seed, if somebody asks you whether it is low phytic acid content or high phytic acid content, you don't have to grow, wait for the seed, perform some biochemical estimation. You can simply do a one-step PCR, resolve on the gel. If you get a similar band, it is low phytic acid content. If you get this type of band, it is high phytic acid content. So such markers we have developed for different other traits also, which are useful for the breeders. Now resource development. Now that we have good amount of genetic resource, we have good amount of genomic resource, we wanted to share it among the scientific community, because millet research community is very small, right? So we developed different online open access databases to share our resources, which are being used by different other institutions also. The most interesting part is gene discovery. Millets also belong to the same family to which rice belongs to. Do you know how much water does rice require to produce 1 kilogram of rice? how much water does rice require to produce 1 kg of rice? It is 2500 liter. 2500 liter means the kind of this black color syntax tank which we install on our home, to produce just 1 kg of rice. Whereas if you take millets, it doesn't require that much amount of water, right? So when we compared the genome sequence of millet with other crops, we found that some around 1500 genes are unique to foxtail, of which 586 genes were annotated as response to water. We thought these genes might have something to do with the water stress resistance in foxtail millet. So we re-annotated them. We found that these genes belong to different gene families. You don't have to go into those details. We performed some qPCR to study their genome, to study their expression patterns. Don't go into those details. What is more interesting is one gene which encodes for a small heat shock protein. When we cloned it and overexpressed it in rice, the rice became tolerant to heat stress. This is the wild type. These three are transgenic lines expressing the heat shock protein gene from foxtail millet. The rice plants, the transgenic lines, were able to survive even up to a degree of 45. Whereas the wild type plants could not survive. You can see that a severe wilting in the wild type plant, whereas the transgenic lines were even surviving up to a temperature of 45 degree centigrade. This story has been published. We have identified similar other genes which are now being overexpressed in rice, where we are seeing the water use efficiency of rice has improved, right? I'm not going into those details. So by this way, we developed a pipeline through which we were able to identify such informative genes, and we published. From the genes that we have published, people were able to pick candidate genes, and they overexpressed it in different other systems. I will show you one example. This ASR4 protein, when this Chinese group overexpressed it in rice, you forget about this side, overexpressed it in Arabidopsis, right? These two are transgenic lines. When they stopped watering for almost half a month, the plants didn't die. And when they watered, the plants recovered immediately. Single gene from foxtail millet, when it is expressed in Arabidopsis, it is able to confer drought tolerance. They performed the same set of experiments in rice also, and they showed that the gene was able to confer drought tolerance in rice. This is another example, LTP, which they overexpressed it in tobacco. They stopped watering for almost 21 days. You can see it looks like all the plants have died, but when they rewatered, the transgenic lines were able to recover. So foxtail millet genes have that potential to impart climate resilience in other crop species also. Right? I'm not going into those details. Then we performed different other studies like comparative transcriptomics, because we are able to see not all foxtail millet varieties or cultivars are tolerant. Within foxtail millet, we were able to see that few are highly tolerant, few are highly sensitive. So we performed a comparative transcriptomics of highly tolerant and highly sensitive cultivars to study how the genes get regulated in the tolerant cultivar and in the sensitive cultivar. And what we mostly look into is the genes, those which are getting upregulated. But here we are looking in a different way. We wanted to see what are the susceptibility factors which can be knocked down in the sensitive cultivar to make the sensitive cultivar a tolerant one, right? But what is more interesting with this comparative transcriptome data is, when we were looking into this data, we found that several genes related to chromatin organization were upregulated in the sensitive cultivar. So we thought that there could be a play of epigenetics in regulating the gene expression, and as we doubted, it was proved that the genes which are responsible for histone acetylation were upregulated in the sensitive cultivar. And what it does is, through integratomic studies, we found that one specific gene is interacting with two other genes, And these three genes are down-regulating the expression of stress-responsive genes in the sensitive cultivar. So when we silenced this gene, this HDA9, through virus-induced gene silencing in the sensitive cultivar become a tolerant one. So that was proved by us, and we have elucidated that mechanism also. In light of time, I'm not going into the details of it, and it is published in the Journal of Experimental Botany. Right. So altogether, if you see, when I started in 2013, very few markers were reported, and only 3 reports were available on the molecular biology of this crop. But with time, we were able to add more markers, and we were able to characterize different genes, gene families, and now beyond genomics and transcriptomics, we are exploring the epigenetics also. So by this way, we transformed a crop which was considered as an orphan in terms of genomic resources into a crop with rich genetic and genomic resources. What is the key? The key is the release of genome sequence information in the year 2012. The moment genome sequence information is available, we were able to do any number of work, right? But if you compare with the animal system or even microbial system, right, plant science is little, what do we call, underrepresented, right? Lot of advanced work happens in those two systems, whereas in plant sciences, it doesn't take off that well, right? Only in the year, after sequencing several species, they sequenced Arabidopsis, right? After Arabidopsis, to sequence rice, it took a lot of time, right? So very slowly, it is taking off. If you could see, a lot of genome sequence that has been sequenced is that of Arabidopsis, followed by rice and barley. And what is more unfortunate is, still 4 millet species genome has not been sequenced, right? And very few millet species have been sequenced, and pangenomics or pangenome information is available for only 3 millets: pearl millet, foxtail millet, and finger millet. In terms of genomics, foxtail millet is doing little better. However, other millets are still neglected. So working with rice, wheat, or maize is very easy because all the omics resources are available. But in case of foxtail millet, right, slowly we are venturing into kodo millet also. Nothing is established. So we always struggle to establish. We know that it is a long way to go, but we are not going to give up, right? And the progress which has been done so far is because of my students, right, who also get demotivated by people saying that don't work on millet, you won't get anything, tumko kuch nahi milne wala hai, right? Still they take up the spirit, and we are able to stand, and that is how the work is being progressed. And thanks for listening. Thank you. Any questions?
Anybody? Thank you, Dr. Muttarasan. Actually, we wanted more time, so you have finished before time and also given very nice talk. I think this is not the days people are not aware of millets. Now everybody is aware of millets. Millets cost more than rice now. Only thing they may not know foxtail millet like that, what millet it is, but everybody it's fashion to eat millet. So if there are questions, I have one question that we can ask more questions from them. You said some 4 species are not yet sequenced. Is it because it is polyploid like that, or it is nobody is working?
Both the reasons, sir. Major reason is that not many people are working. But when I talked to the director IIMR, they said that it is the ploidy.
It is a?
It's the ploidy.
Okay, ploidy level is the problem. Because nowadays it is easier to sequence chromosome to chromosome. Thank you. Please, if you have questions, yeah. Students, faculty, any questions to him? He already told plant science it's not that much getting attention, so you should at least some plant science people should ask some questions. Otherwise, yeah. Rex.
This is not relevant to omics. Basically, you said there is a switch between rice and from millets to rice. That's the period is debatable, but it is long, long ago. The question is, why switch from millets to rice?
Sir,
Is it
I mean,
because of the growing population?
okay.
Nutrition-wise, millets are better than rice. But is it because yield factor or anything else?
Okay. It is not that long ago. If you ask your parents or your grandparents, they will say that they used to eat idli only during Diwali. Right. Once rice was a luxury, whereas millets were cheaper. Major switch came when rice became very much accessible, maybe public distribution system. Second is green revolution, incentivizing the farmers cultivating rice and wheat made the farmers to shift from millets to rice. We started converting the fields to cultivate more of rice and wheat, rice in the south and wheat in the north, leaving millets behind. So if you see the graph after green revolution, rice and wheat goes up and millets come down. So somewhere we lost millets there.
Because of the yield factor, because millets we cannot boost the yield beyond the limit.
It's not that. No, it's not like that. It has different other factors also, but green revolution particularly focused on these two crops. Millets. Why we shifted?
There should be some reason why they promoted rice. There is no politics to promote only rice, like some multinationals were involved. Probably yield may be it was more amenable for increasing the yield. They wanted to improve things quickly to improve the production.
Maybe for rice we would have had good varieties.
Yeah.
Yeah. In case of millets, we just had land races.
Somehow, maybe that is a matter of investigation, I think.
Investigation.
Because in rice also, they found this shortness mutant quickly, that quickly increase the fertilizer responsiveness and yield, probably. Such kind of mutants, even now, millets prices are high because yield is less.
Yield is less.
So that, yeah, thank you. Any other questions, please? If there are no questions, thank you, Tamil. It is very interesting. Yeah. Continue your good work. Thank you.
Thank you, sir. I now request our HOD, Dr. S. Kiran Kumar, to felicitate the speaker.
Thank you. Now we'll move on to the next speaker for the day, Dr. Vinod Skaria, Chief Data Officer, Carnicos Healthcare, Adjunct Professor, IIT Kanpur, Distinguished Visitor, Asoka University. I asked him to reduce 10 minutes of his talk, so I also want to cut down my own time. I want to mention 3 things about him which will be of very interesting to you, especially for students. The first one, many of you come to genetic engineering because you don't get medicine, but he is the person who did medicine and then become a scientist, MBBS, PhD. Very rare in India. So you should ask him why he did that. Okay. So another interesting one is, he is a co-founder of Guardian, which networked the scientists and clinicians to work on rare disease genetics. Okay. There is no disease which can be rare or which can dare to be rare in India because of our population, because the absolute number will be very high. And so in any funding agency, they go by the disease burden, if I understand. So many people should die. Then only they will take up research on that kind of diseases. But he has taken that challenge and worked on rare diseases. That is again very interesting one. The third one, the topic of today's talk, genomics. So he was the person instrumental in sequencing the first Indian genome, whole genome sequencing of Indian. Subsequently, he sequenced Sri Lankan genome and other genomes also. But the first Indian genome, we were all proud, that paper came from his lab. And the other one, he contributed during the COVID, okay? So we were all having COVID, first wave, second wave, third wave, they were all mutating. But these were the people who are analyzing why they are mutating, how they are escaping the vaccines like that. So with these, and if you want to know more about him, he has his own Wikipedia page, so no need to introduce more. Thank you very much. Please, the stage is yours, Dr. Vinod.
Thank you, Professor Parani, for the quite elaborate introduction. I'm humbled, and it's a pleasure to be back here. I think this is my third time I'm coming here. Also a very good chance to meet a lot of my colleagues in science. But of course, I've over the last 1 year moved to a new organization trying to do a bit of things which are very different from what we did in academic science. So the next 20 odd minutes, I will sort of delve a bit deeper into personal genomes. And he really said that we started with one genome. And we look forward to a future where we think a majority of people in this room would probably get their genomes done. Now, why do I say so? I say so because I need to go back into the history, and having learned medicine, I think there is one invention that largely changed the way we practice medicine today, and that was this invention 50 years ago. And anybody can guess what this instrument is? Pardon me? MRI. What else? CT scan. Yeah, that's a CT scan. Now, to put a CT scan in the context, 50 years ago, before the invention of this contraption, the only way a clinician could get into or look into the human body was to essentially surgically open it up, right? And people used to die just because of what we call as exploratory laparotomies. Now you don't die because a clinician wants to look into your body, because they will send you a CT scanner. I last checked in the morning, Chennai has close to 50 CT scanners. And how many of you have got a CT scan in your life? There goes, right? And just think about it. That necessarily means that this guy has really changed the way we practice medicine today. Now, why did I bring this out today? I brought this out because 20 years ago, apart from looking into the human body, we have an ability to look into every one of your 37 trillion cells and the blueprint of the 37 trillion cells and essentially sequence the genome. And of course, the first genome was extremely costly. It costed approximately $3 billion to sequence, multiple different countries to put it together. What really happened in the last 2 decades is what you see here, a price reduction that would mean that a whole genome sequencing becomes affordable as much as a CT scanner. Now, what would that mean? That many of you who would have got a CT scan today, probably in a half decade's time, would also get your genomes done. And that would really change the way we practice medicine. And that's what I'm going to talk about. Now, of course, when we started, there was a big gap. And this is a slide from 2008. That is when I started my lab. And what you see here is essentially the admixture plot of the world. The top panel is the admixture plot of the world, different populations around the world, how homogeneous or how less admixed or how more admixed are they. And the bottom panel is essentially the genomes available in 2008, including the first reference genome, the Craig Venter and the Watson genome, the first personal genomes. And in quick succession, the Chinese, Japanese, and the Korean genomes. And all of them came from very less admixed populations around the world. Now, what you see here in the middle of the map is essentially a population which is quite very large, 1/3 of the global population, which had 0 genomes out from there. And India is right here in the middle of it. That's what we wanted to change. In 2009, we announced the first genome, the 7th country to be able to sequence and announce its own genome. We really did not sequence and stop with one genome. We went ahead and collaborated with quite a number of countries around this region, including the first Sri Lankan, the first Malaysian, the first Philippines, with the founding members of the Pan-Asian Population Genomics Initiative. We created the South Asian Genomes and Exomes databases, and much more recently, the Middle Eastern Population Genome databases. And in 2019, we also completed the Indigen, the first 1,000 pilot genome sequencing initiative. And today we have a 10,000 genome program, which is called the Genome India, which concluded very recently, and we contributed to almost a fourth of the genomes in that particular program. So the question is not that whether we can genome sequence anymore. Its question is about how do we really put it into clinical practice. And one of the places where we can really put it into clinical practice is what we call as rare genetic diseases. I don't like to call them rare genetic diseases anymore because we know that they're not rare. But of course, they are called rare genetic diseases. And to put numbers, there are around 70 million people in India who have a rare genetic disease. 70 million people. What's the population of Chennai? 10 million, 12 million. There's 7 Chennais of people who have a rare genetic disease. And if that doesn't disturb you, I think that should disturb you. Now, the problem with people with rare genetic diseases is they go through multiple physician visits, multiple rounds of misdiagnosis, and lose a considerable amount of time, or a considerable amount of time is rather wasted in the pursuit for a diagnosis, and very few of them can actually get to a molecular diagnosis. And that's really, really sad. And to put these numbers, I would say that if we have 200, 300 people out here, that means 1 in 10 approximately would have a genetic disease. And many of you would not really know that you even have a genetic disease. Now, of course, working about genetic diseases in India is, of course, a big advantage. One, like Dr. Parani rightly stated, that we are a very large country. That means we are approximately a sixth of humanity on Earth. Every rare genetic disease documented ever in literature, we do have them, probably in thousands. And more importantly, we have many more rare genetic diseases which are yet to be discovered, yet to be documented. And unlike our very close counterpart, the other sixth of the global humanity, which is our neighbor, our genetic ancestries are stratified into large and small groups. That is practically because we marry among our own communities, right? Whether by surname, by the caste, by a tradition, by a religion, by relation, we marry among ourselves. And that necessarily means that we have large and small endogamous groups, approximately 4,000 endogamous groups, which encompass a large country, what we call as India. And apart from all this, we still have a close-knit family structure. And like the rest of the world, which has gone through multiple rounds of world war, famine, displacement, India has largely been undisplaced over the last 500, 600, 1,000, 1,500 years, with no major displacement across. That necessarily means that everybody of you would be able to trace your ancestry by 3 generations. Some of them would be able to trace your ancestry by 10 to 15 generations. And that's not very unusual in India. Now, of course, the problem of genetic diseases is that they don't have hope. And this is one such example, probably the first case that came to us. We call him Bhai because he called on the day when the genome was announced, and he called to Dr. Sridhar and said that what you did can actually change my life. And to put things into perspective, he has an extremely rare genetic condition called epidermolysis bullosa. His skin erupts into as if it was burned, it has been burned. And then, of course, it erupts, gets infected, and becomes extremely painful. And he was not alone. He has an entire family of 5 generations, 28 members. I've actually met all of these 28 generations. Over multiple marriages, over multiple visits, we have collected samples for all these 28 individuals. We did arrive at diagnosis, a molecular diagnosis, a mutation in keratin 5, confirming the diagnosis of epidermolysis bullosa simplex. But that's not the end to it. Of course, you can name the disease, but more importantly, with the identity of the disease and the molecular event, you can pretty much eliminate the disease in the 6th generation by offering them genetic counseling and offering them prenatal testing. So you don't have a 6th generation anymore, or for that matter, the curse to this family is put to an end just because you can do genomics. But more importantly, not just because you can do genomics, because even common people like Bhai can understand the importance of genomics and how it can put an end to what we call as a curse to that family. Now, of course, from there, we took it forward to a very large community initiative, today encompassing close to around 300 clinical collaborators from 70 clinical centers, 218 in India and many more abroad. And what we typically do is to work with clinicians who see patients with genetic conditions, put them through a right consenting and a process of sequencing, analysis and interpretation. Whenever we get a pathogenic mutation, we get back to the clinician and say that we have an identity for this disease. and you can act upon it. And that happens approximately in 50% of the cases. The rest 50% of the cases, we step back into the lab to do a lot more research to add value and add evidence so that the clinicians can act upon that particular genetic mutation. I'll just probably discuss just 2 examples. And this one example was from 2015. I vividly remember seeing this news article, and this came in the Indian Express, Times of India, and for that matter, every major newspaper. A family who had petitioned President of India to euthanize 6 out of their 8 children. Now think of the plight of the family who would want to euthanize their own children. And these are the children from the youngest to the oldest in the clockwise direction. They're perfectly normal at birth. At around 3 years, they start losing their milestones. Eventually, they end up in a vegetative state and they die. They don't have a hope. They don't have a diagnosis. They don't have a treatment. And therefore, they petitioned the President of India to euthanize all their 6 children. Now, of course, euthanasia is not permitted yet in India. So they were referred to the official channels for sequencing. We did diagnose the condition as cystic megalencephaly, or in scientific terms, an extremely rare genetic condition called megalencephalic leukoencephalopathy with subcortical cyst type 1. For most cases, this is the end of the odyssey. The patient has a diagnosis. The family would be told that you have a genetic condition. Probably don't marry within your community, but that's the end to it. That's not the end for most of us. We went back to the community because they form a small community called the Nalband. And what we found was even more remarkable. This is an extremely rare genetic disease. The prevalence estimate is 1 in a few million individuals. And what we found in the extended community, around 100 odd individuals whom we screened, was approximately 28% of individuals were carriers for this genetic mutation. And roughly what this number would mean is 1 in every 500 individuals or 400 individuals born into this community would have the genetic disease. Now, apart from a genetic disease, which is 1 in a million elsewhere in the world, now you have 1 in 400 approximately in this particular community. That makes it a public health problem, not a rare genetic disease anymore. So you've taken it from the first case referral in 2016 to the diagnosis, to the publication, to now a diagnostic in the market, widely available for the 2 founder mutations in India for this disease, the Wandernap mutation, which was discovered by Wandernap, but of course patients from the All India Institute of Medical Sciences, and the Nalband mutation, which we discovered and added to the community. And we extensively worked with a number of partners, including the All India Institute of Medical Sciences, Professor Shefali Gulati, NGOs like the Human Welfare Foundation, which takes care of the patient and the extended family screen, and of course, a wide variety of partners in this entire process, right from the discovery to a diagnostic in the market. And today, with the diagnostic in the market, prospective parents can essentially screen themselves and decide on what the reproductive choices could be. Now, this family was very interesting to us because Nalbans, if you understand the history, came from modern-day Mediterranean basin with one of the invaders, and they settled in the ramparts of Agra and Aligarh, and in some cases in the Jaipur. And it's just not an isolated case. There are approximately 1 million odd people today in India who are Indians in almost all aspects, including the language, custom, and tradition, but they all have a genome that comes from a different part of the world. And when they come from a different part of the world, either as friends, as traders, as travelers, or much later as invaders, they all carry diseases which are very prevalent in this part of the world and now manifest in a very different part of the world, and therefore they become undiagnosed in many ways. Now, this is not in isolation. Today, we have a much larger community, what we call as the Indian diaspora, who has moved out of India to the rest of the world. Practically every country today has an Indian. They carry an Indian genome, and therefore they also carry Indian diseases, which may be undiagnosed to the rest of the world. So the point I'm trying to make is that genomics, therefore, is a common endeavor and a collaborative endeavor. There's nothing called an Indian genome that you only need to look at. If you want to help Indians abroad, or if you want to help people who have a different part of the genome from a different part of the world, you need to really collaborate across boundaries. Now, of course, these are good stories to look at because you can make a diagnosis, but of course, that comes with a lot of frustrations, and this is one of such frustrating case. This was a case referred from the All India Institute of Medical Sciences suggesting mitochondrial genetic disorder. We did a mitochondrial sequencing, we did get nothing. The clinician was very persistent, so we did an exome sequencing, and we got nothing. And we were almost looking like stupids because we didn't know how to do genomics. But then at that point in time, we were also doing a whole genome sequencing, and suddenly by looking at a whole genome sequencing, we identified that we had missed out something, and that was this very large deletion in this individual, which is typically not covered in any one of the tests that were available at that point in time. And of course, long story short, this family today has 3 apparently normal children with a founder mutation, which is a deletion, two heterozygous deletions by both parents, manifesting as a compound heterozygous deletion in the offsprings. And you could essentially take it from the clinical study, from looking at exome and genome sequences, looking at patient cells, looking at a mitochondrial assay, modeling that in a model system, and of course making a diagnostic assay for that, so that the entire family of the community can benefit from this diagnosis. And you could do this in the entire workflow in approximately around 9 months, or what we call as a clinical turnaround time. And we have now many more such cases which we have described in this particular paper. If you're interested, a particular paper that we wrote in 2019 with 280 clinical collaborators from across the country, on how we have taken it from diagnosis to research to education to implementation. And of course, today with the policy, we do have a national rare disease policy. And for that matter, most of the diseases that were included in the national rare disease policy came from inputs from our allele frequency and disease comparisons, suggesting that these are not extremely or ultra rare diseases anymore. Many of them are very prevalent diseases. Now, having said that, I will try to close the talk in the next 10 odd minutes, trying to say that we are in a very unique position just because of young people around us. And this is one example that I'll put across. This is a database that we created called the BRCA Indica. And I'm sure BRCA1, BRCA2 is very, very well known to you. BRCA1, BRCA2 are probably one of the first germline mutations on germline cancer predisposition genes that were discovered. And I put numbers, approximately 1% of Indians do carry a BRCA1, BRCA2 mutation. And in breast cancer, in patients with breast cancer, that's approximately 1 in 10. And patients with ovarian cancer, that's approximately 1 in 5. Right? Just BRCA1, BRCA2 would be positive in approximately 1 in 5 breast and ovarian cancer patients. Now, of course, there are many mutations that have been discovered. There are many mutations that have been studied in populations. We compiled practically all of them. And the idea is that, of course, if we compile all of them, the real differences in the annotations between the databases is that they don't follow a uniform nomenclature and uniform annotation for pathogenicity. And that's exactly where young individuals could contribute. We did put together one of the largest reclassification exercises for BRCA1, BRCA2, reclassifying practically all the known genetic mutations in BRCA1, BRCA2, sort of suggesting a final number of what is pathogenic and what is a variant of uncertain significance. And of course, that is not the end to it, because there are many more BRCA1, BRCA2 variants being discovered. And that is exactly where machine learning and computational modeling can actually become very useful. And we essentially did was to train computers on the data sets that we have, which have gold standard ACMG, AMP classifications available. You could use machine learning, artificial intelligence methods. And what we suggest is that you could acquire a very good positive evaluation or an accuracy of almost 95%, suggesting that in future you could probably use this as a very useful screening tool to sift out variants from patient datasets. And because as we sequence more and more genomes, this becomes a very important problem, not just for BRCA1, BRCA2, but for approximately around 3,000 odd genes which have disease implications in the human body. And last week, of course, we have extended this to also looking at breast cancer patients, not just germline variations, but also cancer-associated somatic mutations. This is a work in collaboration with IIT Madras. This database called the Bharat Cancer Genome Atlas was essentially unveiled early this week. This makes genomic data for cancer patients findable, accessible, interoperable, and reusable. And today we have close to around 1,000 odd samples, and we are quickly collaborating with other organizations across so that we can enrich this database. And I'm sure this will become a very useful resource for researchers and including young students like you to go back and sort of sift through and discover new variants and new patterns in many cancers across the country. Now, having said that, I need to step back and say that I said I don't like to call them rare anymore. But the real question is that, I mean, what do you call something as rare and what do you call something as prevalent? And today, the only way people do that is to essentially ask a clinician, right? But having said that, I already said the point that clinicians are very inadept at diagnosing rare genetic conditions because of the very fact that they don't learn genetic conditions much in their clinical curriculum. Now, if you put in a cardiologist, a neurologist, and a pediatrician into a room, and this usually happens in the National Rare Disease Policy meetings, you will never ever reach at a consensus, because a cardiologist will say cardiomyopathy is more prevalent, a pediatrician will say probably muscular dystrophy is more prevalent, and a neurologist will say, for example, epilepsy is more prevalent. And where do you arrive at a consensus? And in fact, there is no consensus ever possible by putting in clinicians in a room. To me, the only way to arrive at a consensus is to probably ask a very simple question. If I sequence apparently normal people like you and me, and we are all, of course, imperfect in many ways because we are all carriers for genetic diseases. And if we know the carrier status of individuals, we can pretty much model our marriage patterns because that's very much known in communities. And we can extrapolate to figure out what is really prevalent in the community. And now, if the prevalence estimate in the community is very different from what the clinicians see, that means that there is a gap. And this gap could either be filled by an accessible, available diagnostic, or the gap could be filled by imparting knowledge back to the clinical community. And this was a hypothesis. And until 2019, we could not really test this hypothesis. In 2019, we had an opportunity to sequence 1,000 odd genomes from Indian populations, cosmopolitan Indian populations. And the idea was that can we really look at the low-hanging fruits in terms of what is really prevalent in terms of genetic diseases, and can we really impact Indian population by understanding this? And of course, today we have a much more richer data set of around 10,000 odd genomes. But having said that, for a billion odd people, 10,000 genomes is too little. Probably we need to sequence a million odd people. And of course, I'm sure that's going to happen over the next half a decade or so. Now, this is one example where we could do this. Easy to say, but very difficult to do. This is where I studied, the Government Medical College in Calicut. So we went back to the clinicians, my own professors, to say that, of course, you're probably undiagnosing or misdiagnosing a certain set of conditions which are very prevalent by the genomic data that is available from that community. And of course, by over 4 years, we could create one of the largest cohorts for this disorder called the primary immunodeficiency disorders. It is one of the largest classes of genetic disorders known. I'm sure you have not heard about it, but a very prevalent genetic disorder. Kids born to this disorder do not have a patent immune system. They typically do not survive the first 4 months of life because they cannot resist the infections that they get in the first 4 months of life, and they succumb to the infections. Now, by making diagnosis available by clinical acumen that could be imparted by making a genetic test available to this community and clinicians, you could actually set up this entire process and actually save large number of kids who could either get IVIG or by hematopoietic stem cell transplant, what is roughly called as a bone marrow transplant, and they could live a perfectly normal life. And this came with support from the government of India through an SCRB grant. It also came from the state government support to establish a very specialized PID clinic and offer the genetic diagnostics at that center. And given the prevalence of this disease, we are today piloting a newborn screening program so that newborns at birth could be screened for these disorders very early on, and you have pretty much 4 months of time to arrange for a hematopoietic stem cell transplant or put them on IVIG. Both of them are today subsidized by the government of Kerala, and they could all have perfectly normal lives. And today we have, of course, one child who was born out of primary immunodeficiency disorder who is completing the 4th year in engineering. The other thing that we can probably do with this kind of a data is to essentially address this question in terms of what should we do for newborn screening. Of course, newborn screening is based on a principle that you have a prevalent treatable genetic disorder. If you can diagnose them very early on, you can sort of treat them so that they can be normal members of the society. And in fact, there are multiple states who are running newborn screening programs. Tamil Nadu has a newborn screening program, very limited number of genes and diseases. And what we show is that practically the genes and diseases which are prevalent in India are very different from the rest of the world, and therefore most of the newborn screening programs might not really have any impact in our ability to save children. For example, Wilson's disease or phenylketonuria or biotinase deficiency should be in the newborn screening programs because their allele frequencies are almost 70 times more prevalent than the Western populations. Then there are, of course, adult onset diseases which are rarely studied and rarely uncovered. Familial hypercholesterolemia is one of it. And we published this paper very recently suggesting that familial hypercholesterolemia genetic frequencies in the Indian population is approximately 1 in 150. That means there are 2 people in this room who have familial hypercholesterolemia. Very treatable genetic disorder, you can treat them with statins, but of course statins also come with what a condition called statin-associated myopathy. So you can also look at the pharmacogenomic variations, and we suggest that approximately 10% of them would require a dosage modification for almost all statins, and a third of them would require a dosage modification for at least one of the statins. So you can do the testing for the prevalent genetic variations in here, and of course the pharmacogenomic variations, and you can actually diagnose and treat these individuals much better. I'll close this session in the next probably 5 minutes. As you see, the sequencing cost is dwindling very rapidly. And that would necessarily mean that we would change the paradigm where we apply genomics in clinical settings. Today, what we apply in genomic settings is that the patient comes to the clinician does examination, history, does a set of labs, and genetic testing is used to confirm the diagnosis of what the clinician thinks is right. But now think of it, if the patient needs to come to the hospital, the patient already has the disease. And if the patient already has the disease, there's very little that you can do for most of the genetic conditions. So the real idea is, can we really change this paradigm altogether? And the change of the paradigm is to actually put this before everybody. In other words, if you can sequence individuals who are apparently normal, look at the genomic variants and evidence for pathogenicity, and start treating them depending upon what is really pathogenic in them. And of course, that is an ambitious story to think so, but not really, because as we see today, the genomic testing is converging. Today, there are multiple kinds of genetic testing, from FISH, RSEGH, PCR, panel sequencing, exome sequencing, so on and so forth. Each of them have their own problems, but today you can do a whole genome sequencing, which can solve all of these problems. Essentially, it's a whole genetic test and more. Whole genome sequencing, of course, offers much more diagnostic positivity than probably any other genetic test that we know today. There's much more information out there. Even we have done this. The idea is that if in the future we could sequence every newborn for very prevalent, penetrant, actionable, and treatable genetic conditions, you can save these children from death and of course, disability in their life. Of course, we are probably the last ones to think. There are many more countries who are doing this. The UK has just started a 100,000 genome program for newborns. And the idea is that you can do a genome sequencing at newborn, and you can actually use this genomic sequence for your lifetime. At different points in your life, you can use it for very different questions that you want to ask. For example, in the middle age, you could ask for hereditary cancer syndromes. Early adulthood, you could look at familial hypercholesterolemia, cardiac channelopathies, which are all treatable genetic conditions. And of course, you can use it for pharmacogenomics to make your treatments much better, much accurate. To put numbers, if you sequence apparently normal individuals, 1 in 20 individuals do have an actionable genetic findings. That means that's approximately 5% of individuals do have an actionable finding. That means that the clinician can act upon it today. And roughly putting the breakup, that is approximately 2% for a cardiovascular disease, 2% for hereditary cancer, and 1% the rest of all genetic diseases together. So take home, as genomic sequencing is becoming affordable. Genomic sequencing can enable disease diagnosis for at an individual level, and put together at a population level can implement public health policy decision making. Genetic diseases are extremely prevalent. 1 in 10 individuals in this room do have a genetic disease, and therefore genomes are for everyone. Genomes are not just for patients who come to clinics. Genomes are also for apparently normal individuals like you and me, and genomic information is for a lifetime. I think I'll stop there and take questions. What I can only say is that as young individuals in this room, you are probably one of the first members of the genome generation, where genomic sequencing will become part and parcel of your livelihood and your life and medical care. Thank you very much.
Thank you, Dr. Skaria, for your exciting talk. And now we can take questions from the audience. There is a question.
Nice talk, sir. So regarding BRCA1 or BRCA2, so there are multiple SNPs they have identified. So how do you prioritize them? Like if the similar multiple genes SNPs, so is it hard or?
Yeah, so the real hard part of it is that you need to reclassify them by the guidelines. And today the guidelines are the ACMG and the AMP joint guidelines. You need to give 28 attributes, or you want to evaluate them by 28 attributes, give a score, and based on the score, you classify them as pathogenic or likely pathogenic. This is a manpower intensive job, right? And that's exactly where Indians can really fare, because if you put in and train students, they can really, really change the way we practice medicine. And no other country in the world can actually do this.
Regarding epigenetics, what is your take on that? Like how this both together?
Yeah, absolutely. See, the point that I want to always make is that while we always talk about epigenetics, right? The basic principles of epigenetics, whether there is a DNA modification or there's a histone modification, is essentially determined by genetic mutations at that locus. Right? A genetic mutation can displace a histone and therefore can manifest as an epigenetic change. A similar way that a splice mutation can change the transcript pattern, the outcomes that we measure, whether it's a proteome or a transcriptome or an epigenome, is inherently embedded in your genomic sequence.
Any students, we have question?
Yeah, the best return of investment is if you can do it as a newborn. But of course, we are all born and we are all old enough. But frankly speaking, as I said, there is no age where you think that it is useless. The best, of course, is in your newborns. And that's why I wanted to talk to this audience to say that we are in an era where probably every one of your children will get sequenced. But of course, adults can also get sequenced, and you will get a 1 in 20 possibility of having a medically actionable information.
I have one question. What would be the cost of genome at the current scenario?
Yeah, this is like asking the question, what is the cost of a city? Depends upon where you go, where you do it, how you do it, etc. The cost of a genome sequencing is typically at this point in time approximately 5 times that of a CT scan, and the cost of an exome sequencing is exactly that of a CT scan.
Any other questions? Okay, if there are no questions, thank you, Dr.
Thank you very much.
for your.
Sir, thank you, sir. Just 1 minute, sir. Thank you for the talk. Sir, I now request HOD, sir, to felicitate the guest, please. Thank you, sir. Now we'll have a short refreshment break for 10 minutes, following which we'll have the oral sessions. The oral sessions will be happening in 2 places. One will be the same Peter Drucker Hall, and another session in room number 003 in the same building. So it was supposed to happen in GN Ramachandran Hall, but it's not there. It's here in 003. So please assemble back immediately after your refreshments in 10 minutes. Thank you.